what I'll do after. All right, so I want to welcome you, Wim, to the reading group, and thank you for sharing a number of really interesting articles, including one that's freshly published in Science. So it feels like a real privilege to be able to talk with you about some, late-breaking findings. I've got a lot of questions queued up, but maybe first, and I've got the PowerPoint slides that you submitted along with that paper to Science that I could queue up if that would help. But if you could just kind of walk us through the evidence. So it looks like what I'm seeing in your cladogram is that it wasn't just like, okay, this entered the mink population and then started spreading amongst the different mink farms, but the cladogram seems to tell a story of multiple independent infections from humans to minks. And then it looked like there was the one outlier of a case of minks transmitting it back to humans. Am I more or less getting the story right?
Yeah, well, I can show a few slides if you're interested, if that
Sure.
helps. Let me see. I can try
make you co-host and see if that helps.
Okay.
I may have to make you host momentarily. Give that a try and see if you can screen share.
It says that I could share my screen, so let me see.
I could also give it a go if
I'll try to share my screen, okay?
Okay, great.
Let me see.
Okay.
Looks good.
I'll
Thanks.
put this in try to put this in presentation mode. Yeah. Can you see the presentation now?
Yep.
Okay. Well, this is a slide. There's a lot of researchers on it, but that's because we are doing this together with a group of researchers who are part of the Netherlands Center for One Health. And there is a lag in this partnership focusing on emerging infectious diseases. And with the institutes that particularly work on emerging infectious diseases, we have formed that consortium to work on the coronavirus outbreak in mink. Mink farming in the Netherlands is not a very big sector, but it has 150, well, it had, I must say, 155 locations. And well, between 1,500 and 2,000 people working there. The animals are fed with slaughterhouse byproducts, especially chicken and fish. The normal cycle, there's a yearly cycle of mink production. It's well, fur production. Their mating period is in March normally, and April, May, the animals are pregnant, and in June, well, in May, pups are delivered, and in June, the newborn pups are vaccinated, and in October till December, the animals are deskinned for their furs. When the outbreak started in the Netherlands with that group, we decided to form that consortium and to do a vast research project in the first farms of the outbreak. And that included necropsies, taking pharyngeal and rectal swabs from the animals, and do take also serum sampling and taking rectal swabs for several weeks. And we also took we did environmental sampling because there was that fear that people might become infected from the farms. And we wanted to exclude that the virus was easily transmitted to the environment. The fatalities, that's good to know. That's also in one of the papers. This is a graph actually from the pathology paper. This shows that in the 4 first farms, fatalities were not extremely high. It goes up to 3%, That can be a large number of animals because in those farms you have thousands of animals. The farms are, in some farms there are more than 20,000 animals. So you can have a large number of animals dying in those farms, but the fatality rate, as when you look at the percentage, is not extremely high. We do have the impression that during the circulation in the farms, that the virus becomes starts to transmit more rapidly and also may become more virulent in the farms. In some farms, we have the impression, although we cannot be completely sure because we sample the farms, and especially after the first farms, the farms were culled immediately for public health protection. So there's no long time follow-up of that. But you have the impression that the mortality stays rather low. When you look at the samples, you can see that in the throat swabs, you see a decrease over time. In the beginning, it was very high, the positive number of samples. In the rectal swabs, it's always a little bit lower. And that has been the same over all the farms. So, well, it's not surprising because it's a respiratory infection. The animals become infected via the respiratory tract. And but the virus is also found, especially RNA of the virus is also found in the fecal excretions. When you look at the pathology of the animals, you can see a severe pneumonia in the clearly affected animals. Animals often have a good body condition, but there is a diffuse, consolidated, and you have dark red reddening of the lungs. And you see also that the other organs, well, I'm not a pathologist, but the other organs are quite normal, but there's a clear reddening of the lungs. So there is a acute severe pneumonia in these animals. And this is a young animal, you see, and these organs in the abdomen are too wet, and that is showing that that there is a clear infection in this animal. When you look at histology, I will not go into this too deeply, but you can see there that this is a staining. The brown is the staining of the virus in the epithelium of the lungs. And for example, in this picture here, you can see that there is a clear inflammation around the alveoli and the bronchi of the lungs. In conclusion, we saw a rapid spread of the virus in the mink farms, highest virus concentrations in the nose, in the conchae, the mucosae of the nose and of the respiratory tract. Number of fatalities, as I said, not very high. Rectal swabs were negative after 2 weeks, so the infection seemed to go through our farm, and the animals did seem to recover, and 90% of the animals developed specific antibodies against SARS-CoV-2. So there is a spread all over the farm of the virus.
And Wim, I think we can only see your first 8 slides. At least on my screen, I see the
okay.
two rows of 4. So if you could slide down a little bit.
I saw that maybe I should. Oh, sorry. I have to switch to.
Either that or slide down. At least on my screen, I can read.
I didn't see that. You didn't see the projection mode. I'll change it like this. Can you see it now?
Yes. Yes.
This is the slide of the all the sequencing that we did. What we did, we sequenced all the isolated virus in the different farms, and what you can see, well, this is a phylogenetic tree, and it shows the relations of the sequences, how related the sequences are. And what we saw is that there were, and it's on top in the you can see that in the coloring, there were 5 clusters of viruses in all the mink farms. This is a picture of about 16 farms. And that stayed the same all over the outbreak. So in the end, we had outbreaks in 70 farms, and they were still in those 5 clusters. So the idea is that there were 5 introductions in the farms from the human field. So human virus spread to the mink, and between farms, it was spread after those 5 introductions in 5 clusters to all the other farms. And well, important. This is from the a paper of Bas Oude-Munnik, where Bas Oude-Munnik was the first author. And what you could see is that in that paper, it's described that when we started testing all the all the people working in the farms, and we tested them by PCR and by serology, and we found that in total, of the 97 people tested, there were 66 people found positive either by PCR or by serology. So 66 people working in the farm were infected. And when of those 66 people, we were able to obtain virus isolates of 7 people, and we sequenced those viruses. And these viruses were always related to the mink. So it was clear that the 7 people had obtained the virus from the mink. And also for the other remaining number of people, of those 66 people, it's very likely that they obtained the virus from mink, because this prevalence is very high. And all the sequences that we were able to evaluate were connected to mink. So it was clear that people were infected from the mink farms. Also, in the disease histories, these people always attracted the infection after the infections were spreading in the farms. So that was a very important reason for the Dutch government to cull all the farms and to continuing the culling of the farms for public health protection. Well, there was next, there was an important question. How dangerous is this? Does this spread to other animals? So we started testing cats around the farms, and we found positives. Also, we found a positive in a dog on a farm, and 1 bird was tested positive from taking a feed swab. And we are still in, there's ongoing work in testing other wildlife around the farms. But up till now, our impression is that these animals have contracted the virus from mink, but do not seem to spread this virus to other animals or to people. But additional testing of wildlife is still ongoing. It's important also because when wildlife gets involved or pets get involved, there may be a threat, or again to a threat for public health, and especially for people working on the farms or living around the farms. So in conclusion, SARS coronavirus is highly infectious for minks. Minks can transmit the virus to humans, back to humans actually. Especially farm workers are exposed. So after the outbreak, all farm workers were advised to wear personal protection. Minks may act as a reservoir for the virus. So that was the main reason for the authorities to cull all the mink farms. Actually, mink farming is stopped now in the Netherlands by the 1st of January. It's not allowed to do any mink farming anymore. That was a decision already earlier, because there were more and more complaints about the ethics of fur production. And so there was already a decision that mink farming in the Netherlands was to be phased out until 2024. And actually, the government has accelerated that decision and brought that to the 1st of January of 2021. So all mink farms are now closed, and all farmers receive compensation from the Dutch authorities because they have to stop their farming. We are also looking at SARS coronavirus 2 in the environment. We took, as I already explained, we took air samples. that was always negative around the farms, outside the farms. We found positives inside the farms, and we still are working on testing all the manure from the farms. We have found already, and that's a paper in press, that the virus is not very stable in mink, but the RNA of the virus can survive for quite some time in mink manure. So there is still research ongoing what should be done with all the manure from the farms. This is the consortium. As I said, we have still a lot of research work ongoing, and that focuses also on the new variants, because we find those new variants in minks also. And so we are testing those viruses, whether they're more virulent or more easily transmitted between animals. So that's still a lot of work, and we are still expecting at least 10 more papers on these mink outbreaks in the Dutch farms. So that's for a short introduction, and I guess if you've seen the papers, you've probably some questions. I'll try to unshare this. Let me see. Stop share. Okay. I'm happy to answer questions.
Thank you, Wim. That was a wonderful, really informative presentation. And if anyone is joining for the first time today, feel free to just unmute yourself and ask a question, or type the question in the chat first, and then we can call on you. So Wim, one question. Early in your presentation, you said that there might be some evidence of increased virulence, but is there? It seemed like the science paper was saying that it did spread, to some people that had direct contact with the minks, but the mink strains weren't spreading more broadly in the human populations. Do you think there was, is it increased virulence that you were thinking about just within the mink populations themselves, or do you think it would have increased virulence once it crosses back to humans?
Yeah, well, we are looking, what I was talking about was really in the minks. What we saw in the Dutch situation, that people, other institutes have been doing sequencing of all the human infected people around the farms also, and only the people who worked on the farms or lived on the farms were showing sequences related to the mink. That situation was different in Denmark, where there was also spread of the mink variants into the human population. And the main reason is probably because after the first few farms in the Netherlands, we decided to cull, actually the authorities decided to start culling the farms, because we saw that all the young animals, the newborn animals, were infected also. And the idea was that it was too much of a virus spread to control without any threat to the people living around the farms. So that's why we started culling the farms immediately. And that's probably the reason that the mink variants didn't spread any further in the human population in the Netherlands. Whereas in Denmark, they decided to keep the farms, and they sort of hoped that the virus, that the animals would recover again. And that was all likely to be true. But in the meantime, the virus was spreading to the people around the farms. And that's why when they saw there were also new variants spreading to the people around the farms, they said this is a situation we can't control, and this is becoming too dangerous for the people outside the farms, living around the farms. And that's why they decided to cull the farms also in a second assessment of the situation.
And it seemed like one of the ending points you made was that it's still an open question how it was getting around the different farms. I think in the paper you talked about some of the farms being owned by the same person, and that was sharing the same strains. Were you seeing other kinds of movement between the farms other than outside of, a particular family company?
Yeah, well, in some farms, well, as you said, some farms had the same owner. And there was clearly an impression that people were going from one farm to another. And there were also farms where people worked on more than one farm. So people working on, well, it's also, we have seasonal employees also on those farms, because especially around the delivery of the young animals, and after that of the young animals, there is also a vaccination campaign in those animals. And that's a very busy period in those farms. And that's why they have seasonal workers coming to the farms. So there were also foreign people, like people from Poland, coming to work on those farms. And it's likely that that was a risk factor for virus spread between farms. But this is still under investigation. We have done questionnaires on all the farms about their contacts, about their farm management, about their hygienic protocols, etc., etc. And that's going to be a paper, and we hope that from that paper it will become more clear what is the main risk factor and what could have been the main transmission mode of the virus between the farms.
I also had one question on the cladogram that you showed us right now in the PowerPoint. It looked like some of the points of divergence were actually probably before SARS-CoV-2. So the question in part is just about the timeline of when SARS-CoV-2 arrived in Europe. And it seemed like some of the timeline that was at the bottom of the cladogram seem to imply that the divergence between those different strains happened like well before like March or February.
Oh yes, you see some of the strains that have become divergent in quite in the beginning, and you see also strains that become divergent later. And so that's also something we're going to analyze further. You can do a sort of biological clock calculation, how quickly the virus evolves, or how quick the evolution of the virus goes in the mink farms. So that's also part of the research that we're still doing. And the idea is that when you use that information from the sequences and the dates of infection, you can sort of calculate back to where the first strain was introduced in the farm or in the population. So that's work we're also doing. And the idea is also, if you do that for the different farms, it may also become clearer how the spread between the farms was taking place in this huge outbreak.
Thanks. I've got lots of questions, but if other folks want to jump in, please do.
Yeah, I had a question. So I was wondering if you'd care to comment on differences between how the coronavirus motivated culling in the mink farms, how that differs from bird culling in poultry farms to prevent the spread of the avian influenza. Because to me, so in India we have the bird flu as well as the coronavirus spreading, and to me it seems like in the case of the coronavirus, because the pandemic, it's so much a human concern. It's not so much the body of the animal that's targeted as much as this worry about the spread of the virus. Whereas in the case of the bird flu, the culling seems to effectively terminate the animal body and therefore also terminate the spread of the infection. So I was wondering if you could talk about if there are any differences.
Yeah, I indeed think there is difference between avian influenza and SARS coronavirus. In SARS coronavirus, the reason to cull those farms is really driven by the public health threat. In the animals, well, you see some severe infections in the animals, but as I showed in the graph, the fatality rate is not extremely high. So also in the first farms of the outbreak in the Netherlands, we followed that quite closely. And what you see that indeed the animals, after you have 2% of the animals dying. The rest of the animals in the farms seem to recover quite well. So for the animals, you could still continue the farm, but the reason really for the authorities to start culling those farms was that the virus keeps circulating in the farm, and there will be an ongoing threat of the virus going from the animals to people, and that's why the authorities decided to cull the farms. In avian influenza, that's indeed different, because the fatality rate of poultry poultry in, for example, pig production. How I say poultry production, the fatality rate is extremely high. It can be over 90%. So there's also an important veterinary reason to cull those farms to avoid spread to other farms. In the end, avian influenza can be a zoonotic virus too. In some highly pathogenic strains like H5N1 is also a public health threat. But the primary reason is to cull the farms for veterinary reasons, to make sure that poultry production is not too hardly affected by these outbreaks.
I was wondering if you could also just comment. It might still be early days because the outbreak was much more recent. But what happened in Denmark and I guess throughout other Nordic countries? I understand that it's SARS-CoV-2 is continuing to spread. Is there any sense of are these completely independent lineages from the ones that you've been studying, or?
Yes, the lineages in Denmark are different from the lineages in the Netherlands. What you can see if you, well, for example, if you take all the lineages from people as well as mink, you will see that the mink sequences in Denmark are close to the human sequences in Denmark, and the mink sequences in the Netherlands are closer to the human sequences in the Netherlands. So there is really a clustering, a geographical clustering of the different sequences that you obtain. What we saw in other countries in the mink production is that in most countries there were introductions also, and there were large outbreaks also. As far as I know, for example, the mink production, although they only have a few farms in Belgium, for example, they stayed free of the virus. In Denmark, mink farming is much bigger than in the Netherlands. Denmark is the first mink fur producing country in the world, and it's 5 times as big as in the Netherlands. And that's for one, that was a reason to not cull the farms immediately, because mink production is far more important in Denmark. But in the second phase, because they didn't cull the farms, it became much bigger, because it was impossible to control it any longer after the virus spread to more farms and also spread to the human population. So that's the reason why they, and in Denmark, they run into other difficulties also, because there was a development of a new strain that seemed to be more infectious for people, like the one that's now discussed for the UK in the human field. And they also run into the problem that they couldn't deal with all the animals that had to be euthanized and destroyed. And they started burying those animals. And by burying those animals, the virus was sort of brought into the environment, and that could lead to other complications again, because you might have contaminations of wildlife, and also again via wildlife spread to people. So there was a really difficult situation in Denmark. It was sort of becoming a disaster, and I still see that they have a lot of problems in controlling all the problems around it.
I was just wondering, just
directly related to that, if you could speak a little bit more about the process of, not really the process of culling, but what the differences between what countries choose and what it means kind of infrastructurally also. So what happens after you have a mass culling of animals in this particular instance, mink?
Well, the normal when you have the culling of a farm, the normal procedure is that well, there are different methods to be used. For example, in poultry production, it's possible to close all the air inlets in the farm and to use CO2 to euthanize the animals. In mink farming, that's very difficult because these farms are often have open walls on the sides. It's just a roof, and all the cages are under a roof, and there are no walls in the farm, in the stables. So it's not possible to use that method for the same method for mink, and that's why they use those gas boxes to kill the animals. And that's far more laborious than in poultry production. And the other problem is that you need, when you are culling a lot of farms, that you need very large incineration capacity. And for example, in the situation in Denmark, that was not available. And that's why they started burying the animals. And that was causing a lot of problems because the animals are infected, and you get other infections in the animals also, and a lot of gas development in those bodies. So it was a huge problem there. So it was not a good choice of the Danish government to use that kind of methods. It was what they could have done, but that's also depending on capacity. They could have frozen those animals. That would be a better solution, and then take them to incineration afterwards. But if you have so many farms where you're culling animals, that there can be a real logistic problem in how to deal with that.
Intense images, kind of.
Yeah, well, that's something you normally would not like to think about, but if you have that situation, it's not very easy to deal with.
Yeah. Julieta, you had go ahead, Lyle, if it's right on that.
No, actually, I think yes, the other question is still on that topic, so I'll come in later.
Okay, great. Julieta, go ahead.
I'm muted because there is a 4-year-old screaming around me. So like a question that I have is like looking at the culling of animals directly involved with SARS-CoV-2, like the pigs in Germany, for example. Tonsis, I think, was the pig plant that was that had a big outbreak in Germany, and just like thinking about like the importance or like the meaning of the animals, like specifically the minks as being associated with wealth and having like this kind of symbolic value, and if that makes any kind of difference in how the animals are dealt with and for the culling process.
Well, of course, the number and the size of the animals is important there, but also the impact or the economic importance of the animals make a difference also. Mink in the Netherlands are not important for food production. They're not used for food production. In the Netherlands, mink production is sort of seen as less important and not accepted by all people as a sector for ethical reasons. A lot of people think that you shouldn't kill animals for their furs. Well, that opinion differs in a lot of countries. In especially in Eastern Europe and China, it's more accepted to use fur from animals. In the Netherlands, that was a reason to stop mink farming by 2024, but that has been accelerated due to the coronavirus crisis. I indeed think when there is culling of, well, there is a need to cull, for example, cattle or pigs, that would be an even more a higher impact decision in our region, but the difference between countries, of course.
Lyle, go ahead.
So my question, and I'm sorry if you addressed this at the beginning, I missed the first couple of minutes, but my question is about the fact that it seems that the virus spreads much more easily to mink and among mink than to many other animal species, although there's been occasional cases, but it's been not such large scale outbreaks as in this case, and not the reverse reinfection to humans as well. So to what extent does this provide evidence of the possible zoonotic origin? Possibly mink farms in China, something like this. And is there, so what kind of degree of evidence does this provide for that?
Well, there is a difference between animal species, how susceptible they are for SARS coronavirus 2. And there is also a difference in the level of demonstrating clinical signs due to SARS coronavirus 2 infection, and also a difference in between animals in how, to what extent they spread the virus to other animals. So excreting the virus by exhaling the virus with their coughing. It's clear that minks are very susceptible, but also other martyrs-like species, like mustelid species, like ferrets and martens, are also susceptible to SARS coronavirus 2. So it is possible that these animals can play a role in the transmission of the virus to humans. And we know that SARS coronavirus 2 originates from bats, because there is an ancestor of this virus found in bat species in China. And it relates also to SARS coronavirus 1, where these ancestor viruses were also found in bats. But the point is that in these bats, the virus is still rather different from the strains that we find in humans. And it's questionable whether these bat viruses, the original bat viruses, are really infectious to people. So the idea is that it is likely that there has been an intermittent species between that transmission to people. And that's still under discussion. Actually, there is an investigation group of scientists now in China of WHO looking into this. But well, people think that there may have been an intermediate host. And it is true that, for example, mink could have been that intermediate host. If there was a mink farm infected in China, for example, from a bat, that it is theoretically possible that minks could have been that intermediate host. But it's also possible that other species, that it is another species that we don't know of. And this can be a very exotic species. There was a discussion about pangolins, because there is a virus like SARS coronavirus 2 also circulating in pangolins. So these pangolins, well, their scalps are eaten sometimes in China. Well, I hear from Chinese that it's not a very regularly eaten food, but it is possible that some people have kept or have traded those animals for their scalps. And so one of those species could have been an intermediate species in this, well, before this became a pandemic.
Yeah, just quickly, interesting, because you were noting and the growth of the mink industry as well. So in China, and
yes,
so
in
some
China,
other maybe kind of economic
In China, mink production is important, and also they are keeping raccoon dogs also for their furs. And it has been shown that raccoon dogs are also susceptible to SARS-CoV-2. So, but I think this is something that we will have to closely monitor in future, because if those species stay susceptible, and we may have maybe a lower circulation of coronavirus in the future after vaccination, it may still be a threat for public health when these animals become infected. So it's something that we will have to monitor in future.
Nina, you have your hand up?
Yeah, thanks. Thanks for this presentation. I had two related questions about your phylogeny. So apologies if you already answered these. You said, I think, that 7 of the sequences were obtained from the reinfections of humans. I was wondering if those are significantly different from the other human strains that had been circulating, if they had undergone like a stepwise evolution in the mink that made them significantly different. And if you think that if you have any evidence that they've been sort of circulating more widely in the human population, if they become part of the human soup of COVID, or did it seem like they were not transmitting beyond those first contacts at the mink farm, if you have any evidence?
Yes, of course, we do quite some sequencing in the Netherlands, but the number of isolates from the human population that are sequenced is still far below 1%. And that's the same for most countries in the world, except for the UK. In the UK, they are doing a lot of sequencing, but in most countries of the world, the number of sequenced strains is lower than 1%. But what we saw from those that sample size also in the Netherlands is that the infections that we saw in the people from the mink farms, so these were people either working or living on the mink farms, these sequences were closely related to the mink, but these sequences didn't spread to other people. And that's probably due to the fact that there were very strict regulations around the farms after the outbreaks were detected. And there was, by that time, there was a lockdown of for people also to stop the spread in the human population. So we were really, I think we were really lucky that we didn't see any further spread. In Denmark, that was completely different. In Denmark, the isolates from the people who were infected from the mink farms were really spreading to other people also. So that's why the situation in Denmark became much worse than in the Netherlands. And in the Netherlands, it's probably because we made that early decision to cull the farms as soon as they were infected. So we started a surveillance program also in the farm. So all the animals that were dying in the mink farms were tested for SARS-CoV-2. And when there was a positive found, all the whole mink farm was culled. And there was only 1 day in between. So one positive detection, the next day the farm was culled. And since the outbreak didn't spread that rapidly, we were able to keep that pace. So to cull the farm right after they were detected. And that's probably the reason that the spread to the human population never became a problem. But we have been warning other people in other countries where mink farming is a practice, that it was important to follow the mink farms closely in the surveillance. And that's probably also the reason that in Belgium, for example, our neighboring country, that's probably the reason that they were able to keep the virus out of the mink population. And the mink after the furring of the animals, they didn't have any infected farms there. So I think also for other countries where mink farming is still going on, we still are warning them, put a good surveillance program in place to make sure that whenever there's infection, you can take measurements within a short time.
Thank you. And if I could just follow up with a question, so you said that the sequences that were found in humans were related to mink. Is the molecular difference between the ones related to minks and the ones that came directly from humans so great that it could affect vaccine efficacy, do you think?
I didn't hear the last sentence you said.
So it sounds like there's been genetic change between the viruses that go directly human to human or from mink to human. And I was wondering because I can't really tell from looking at the phylogeny if that's a great genetic difference or very small and just enough to identify them. Does it seem that it's great enough to affect vaccine efficacy if it were to get back into humans?
No, the changes are too small.
Okay.
The virus stays infectious for people. There are just a few mutations in the viruses. I had heard that suggestion before. If you have an attenuation when the virus adapts to the mink population, it may become less infectious for people. But our impression is that all the viruses that we saw in the mink farms were still so close to the human sequences that the virus would still be infectious for people.
Okay, thank you.
Yeah.
Super interesting. I've got a million questions, but maybe I'll go back to the one that's related to Lyle. So I think your science paper does a fabulous job of also just kind of bringing us up to date on the secondary literature about all the other species that have been sampled thus far for SARS-CoV-2. And, you know, back on the point about the bat coronaviruses, this paper also makes a clear point that, you know, there is a common ancestor, but it's probably has diverged somewhere between 1948 and 1982. So this has probably been rattling around somewhere for quite a long time. One of your other papers, I guess it's let's see, I think it's the one that appears in Veterinary Pathology reports, and you mentioned this a little bit just now in the presentation, that you were sampling feral cats. And in my mind, that's, you know, one animal that people all over the world have interactions with. So I guess in your study, you found antibodies in a number of cats, you know, RNA in one animal, but not enough to sequence or characterize. But maybe it's a question also about the secondary literature. What is your sense of how well SARS-CoV-2 has been studied in domestic cats?
Well, there is quite some work ongoing in animal studies. There is actually a WHO working group which has a meeting every week where all the new animal studies are presented. So that works very well, and it's also very good because of course we have a lot of questions about what species are susceptible. And the second question is also very important. What role do these species or these animals play in the epidemiology of the virus? And well, that relates, of course, to the public health threat also. So it's very good that there is this WHO working group, because people show what they're doing in the different animal experiments. And by doing that, there is a far lower risk that people are doing the same experiments, and that's also good for well, you don't want to use too many animals, at least not more animals than really necessary. So it's important. It's very good that this working group is there, and what we see there is that there are quite some interesting studies. We have more and more information about what animal species are susceptible. And you see also huge differences between animals. For example, we see, for example, in hamsters, that these animals are showing clinical signs and are very susceptible and also showing clinical signs. And that makes that species very suitable for vaccine studies. And that's what we're doing here also. Well, vaccines have to be studied in animals before they can be used in people. So we are doing a lot of vaccine studies here in hamsters. But you see also animals where you see that when they receive a large dose of the virus, that it causes some infection, but it does not cause any spread. And an example of that is, for example, pigs and even cattle. Some infections have been shown. And but these animals do not seem to play a role in the epidemiology, and that's important information, because when species that we have very much around us, like for example pigs, are in a lot of countries there are pigs, and pig production is very important for food. So it's very important to know that these pigs are not so susceptible and do not play a role in the epidemiology. And so there is a difference between all, and we actually are doing studies at the moment in cats. We are looking at cats in households. In households, we see that cats become infected, and we are now modeling based on the number of infections and the time that they're excreting the virus, whether these animals really play a role in the epidemiology. I don't have the final results yet, but it looks like cats are susceptible in households, and cats become infected, but they do not seem to spread the virus to other cats. So that's important to know, because a lot of people have cats in a lot of countries. And so it's very important to know whether these animals can play a role in the epidemiology. And maybe also, this may be a changing situation. It's also possible when new strains develop that species become more susceptible. So this will be something to keep following in future also. And so when we discussed it, there is probably a lot of things, aspects of this virus that will keep us busy for the coming years.
Go ahead, Sriya.
I have a question that moves away from cats and back to farms again. But I was very curious about whether there's, if all the decisions are kind of taken by the nation, or if there's some sort of, especially in Europe, is there a kind of EU policy? Because so much of this depends, not depends, but so much of this can mean transmission between countries, whether it's from seasonal workers going back and forth and so on. So I'm just curious about what kind of regulations there might be in place, or if there's been conversation about doing this separately, and if that's okay, or if there's some sort of more communal effort to think about these.
Well, when it comes to protection for zoonotic diseases, the final decisions are mostly, well, it's still made on a country level, but there is more and more interaction between countries. And the organizations that play an important role there is the OIE, that's the International Organization for Animal Health, and also WHO. When the WHO made a report about the outbreak in minks also, and OIE is giving advices on doing surveillance and monitoring, not only in minks, but also in other wildlife. So these organizations try to connect what decisions and what research is being done in the different countries. So it's very important that these, I agree that it's very important that these organizations stay active on these questions for this virus. Yes.
Yes, and can I also ask a question? It follows up right with what Sriya just asked right now. Can you speak a little bit more about the legal basis? Because in the BBC News regarding the culling in Denmark of the mink, the authorities worry that a mutated form of the coronavirus could actually hamper the effectiveness of a future vaccine. And this is something that Nina already mentioned as well. So it could lessen the effectiveness of that vaccine, but it wasn't sure at the time. So the question is, the radical step of culling all these animals, is this appropriate always when human life is endangered, and not only with mink, but also with pigs and cattle or birds? Where is the cutoff? And are also animal welfare organizations consulted? You mentioned the OIE. is important, or the World Health Organization, but can you speak a little bit more about the legal basis? Because the decision in Denmark was taken very fast, and also it might have issues now with contaminating the water. Speaking to what you mentioned earlier, that they have to be buried, it would have been better if they were frozen and then burned later. So this seems to have been a very fast decision. So I would be interested if you could speak a little bit more about the legal basis of this.
I think this is a very good question, and also a very important matter, because when you take a harsh measurements for public health protection, and in this case, this is a virus that is causing a pandemic in people. So you have to make decisions for public health protection. But on the other hand, when you take such harsh measurements for a certain farming practice, the measurement that you take has to be proportional. So you cannot simply get rid of all the animals simply because of public health protection. Right. And also these animal production farms, they have rights also, and they will go to court if such a measurement is not proportional. And that was the problem that the Danish government was talking about. They were taking a measurement for public health protection, but the farmers complained that there was no proof that the virus was so dangerous. And that's difficult for authorities. Sometimes they have to make decisions because before they have all the information they need from research. So on one hand, it's important to do good research and to try to do that as quick as possible. But you can run into the situation as an authority that you have to make a decision without having all the information that you would like to have. And that was happening in Denmark. And I agree, this is difficult for the authorities, but sometimes they have to make this kind of decision. And as a researcher, we try to find all the information, or try to give all the information that we have on research to our authorities, and they also give us research money to do that kind of research. But sometimes you still have questions that you cannot answer. For example, in the Netherlands now, there is a discussion about, I had this morning another meeting discussing what should be done with all the manure from the farms, because we don't know whether all this manure is really virus safe. And normally the manure is brought onto the land as a fertilizer, but that could be a risk when there are still infectious particles in those manure outputs. And so that's the discussion: what is safe and what can we do to do that as safe as possible. And again, the authorities will have to make a decision there without having probably all the information that they would like to have. And well, I agree. Research doesn't answer everything, and sometimes you have to make more decisions than you would like to take, but they still have to do that. So this is really an important point.
Thank you so much, Wim. This has been a lovely hour to spend with you. Just briefly on the question about vaccines and immunogenicity, I'll try to summarize some stuff that I've learned from the primary virology community. So the fact that something might shift subtly on a surface of a virus is unlikely to make it escape a vaccine. So these are most of the vaccines that are being rolled out right now are generating polyclonal antibody responses, which means that it's not just one molecule glomming onto the surface of the virus, but it's multiple. So the same questions about the mink virus are now being asked about the UK variant strains, whether they're going to escape the vaccine. But the answer is probably no, just because even if part of spike changes, there's going to be other parts of spike that the immunoproteins are going to stick to. But it's all very complicated, very early in the game. you know, at least my takeaway, both from the Denmark case and the current UK debate, is that the sky isn't falling. I mean, the sky is falling. We're in a pandemic. You should wear masks and socially distance and do all those other things. But you know, at least the consensus amongst the virologists, they don't seem to be. I almost sent out a message on Christmas after hearing some of these news, you know, saying, don't worry, the sky isn't falling any more than normal these days. But thank you very much, Wim, and I hope when your paper on cats publishes, you'll come back and share it with us. I'd really love to hear your latest once that's available.
I'll be happy to do that. So well, it's very nice to talk to you, and good luck with the group. It looks very good and very interesting.
Thanks so much, Wim. And a brief plug for next week, Tom Van Doren will be discussing Deborah Bird-Rose's work on bats. And Rachel, do we
okay, that would be very nice to join also, yeah.
Yeah, Rachel, do we have a time for next week?
We don't have a time yet. I've got an email out, so we'll see.
Okay.
Yeah, we send an email out. I'll be happy to join again.
Wonderful. All right. Thank you, Wim.
Thanks, Wim.
Okay. Thank
Bye,
you.
everybody.
Bye-bye.